students, for 10th standard we are going to study about the structure of the chromosome, and that part is actually the last part of the chapter on cell division. Now all of you know that the chromosome resides inside the nucleus. This is showing the nucleus, and you have the chromatin material. So the chromatin material is actually the chromosome, which is not in the condensed form but in form of thin fibers. But if you see the structure of this chromatin material in a high resolution, that means if you are magnifying it, then you can see the DNA strands, which is a part of the chromosome. They are wound around in certain places like this, and this molecules, this structure is called the nucleosome. And this nucleosome has 8 molecules of the histone protein. Of course, this is found only in the eukaryotic cell. So 8 molecules of histones are there, 4 are seen, 4 on the reverse side will be seen. So this is the DNA structure with the nucleosome. Now, if you see, there are certain, you can see here that along with the histone proteins, there are certain non-histone proteins are there. So a chromosome is a highly condensed structure which contains the DNA, which is the basis of all the genes, which are actually the nucleotides, which are polynucleotide chain, and there are various proteins which are attached to it, and that makes a supercoiled structure of the chromosome. Now all this is inside the nucleus, the condensation of the chromatin into the chromosome. So this is a chromosome. You can see this part is called the primary constriction, and the centromere region, there are 2 chromatids. This is a single chromosome with 2 sister chromatids, and these chromosomes have the location of the centromere can differentiate the chromosomes in different form. Like though that is beyond your scope at this 10th standard, but they have different types of name like metacentric chromosome, submetacentric chromosome, acrocentric chromosome, telocentric chromosome, on basis of the position of the centromere. And beside this primary constriction, there can be a secondary constriction. So you can see the chromosome here is a metaphasic chromosome with quite a thick structures, but the DNAs are wound here inside this chromosome. Now this is the centromere, which I have already told you. So the chromosome in a highly condensed form that looks like this with alternate light and dark bands. The light bands are called the euchromatin area, and the dark bands are called the heterochromatin area. Now, this is the type of the different type of chromosome. If the centromere is in the middle, it's called a metacentric chromosome. It is somewhere little bit towards one side, not exactly in the middle, then we call it a submetacentric chromosome. Almost near the terminal end on one side is called an acrocentric chromosome. And if the centromere is terminally placed, then it is called a telocentric chromosome. Besides this, the centromere may not be present in some cases. We call it an acentric chromosome. So this is the centromere. Okay, the centromere is the region where the spindle fibers are attached. Okay, and you can see here the different forms of the chromosome shown in details little bit. Now, so the chromosome with the secondary constriction, you can see this is the primary constriction, this is the secondary constriction. Above the secondary constriction, a region is there. This is called the SAT chromosome or the satellite chromosome. These chromosomes are found in human chromosome in 13, 14, 15 and 21 and 22 pair of chromosomes. We find this type of the SAT chromosomes. Now we'll go in the details of the structure of this DNA. So the DNA is actually a polynucleotide chain here. You can see there are 2 chains. We have shown in 3' and 5' direction, and this is also 3' and 5' direction. This is 3', 5' you won't understand. This is actually the attachment at the 3rd carbon atom of the sugar and the 5th carbon atom on one side. So it's called 3', 5', and the attachment of this phosphodiester bond on reverse will be the reverse on the opposite side, so that we call it 5', 3'. So you can see both are anti-parallel. That means the orientation of one of the strand and the other strand, the location is just opposite that shows the DNA, the two strands of the polynucleotide are anti-parallel. So from one point to another point, this is approximately, means where they are wound around. This is called distance is about 34 angstrom, and in a bDNA generally there are 10 base pair in between these 2 points. So that means this attachment between these 2 base pair is approximately 3.4 angstrom, because there are 10 base pairs. So 34 divided by 10. So in the distance between these strands of the nucleotide is 3.4 angstrom, but it can differ in different cases of different types of DNA. Now the DNA, what do we have? In the DNA we have a N2 base. This N2 base is called the nitrogenous base. There are 2 types of nitrogenous base, purine and pyrimidine. In purine you have adenine and guanine. In pyrimidine you have cytosine and thymine. Of course there is another pyrimidine base which is not found in the DNA called U or uracil. This is found only in the RNA. So I have not mentioned it here is because in DNA you have these 4 AT, usually the complementary pairs, and GC is the complementary pair. So one purine always complements another pyrimidine or vice versa. So there is a sugar. The sugar molecules are there. There is a pentose sugar. The sugar molecule structure like this. Now here you can see 1, 2, 3, 4, 5 carbons are there, so we call it a pentose sugar. Pentose sugar is also called a ribose sugar. You can see this is the structure. Need not study the structure, but I am showing here the position of the carbon atom. So this is a 5 carbon, 1, 2, 3, 4 and 5. So 5 carbon sugar, so it's called a pentose sugar. Beside this you have a phosphodiester bond. That means a phosphoric acid is tying the 3' and 5' end of the sugar. So on basis of that we call the strands as 3', 5' and 5', 3', just anti-parallel strands of the 2 strands of the DNA. Now here what is a nucleotide? You have to know this. Many times in ICSE this question has been asked. A nucleotide consists of the phosphoric acid, the sugar and the base, nitrogenous base. So N2 base, the sugar, okay, the deoxyribose sugar and the phosphoric acid, this is a nucleotide. If you don't have the phosphoric acid, then we have only the sugar and the base, nitrogenous base, then we call it a nucleoside. And their names also differ. When you write, like say for example adenine and sugar, we call it adenosine. Adenine, sugar and phosphoric acid, we call it adeninic acid. This is a nucleotide. Here we have shown the 2 strands of the DNA. You can see the bases are attached or on the DNA like this, and they are always complementary. A will be complemented by T, T will be complemented by A on the 2 strands which are anti-parallel. If it is C here, it will be G here. If it is C this side, it will be G on the opposite strand. So this is the basis, and each of these triplet codes generally they code for an amino acid. So we call this the codons of the different types of amino acid. This is of course from the DNA, it is transcripted into the mRNA. Now we have to know a little bit about the RNA. Since we have studied about one type of nucleic acid, we have to know a little bit about the RNA. RNA stands for ribonucleic acid, as DNA stands for deoxyribonucleic acid, RNA stands for ribonucleic acid. And there are 3 types of RNA. One is called the mRNA, the messenger RNA, T is called the transfer RNA, and R is called the ribosomal RNA. Usually the rRNA, this makes the ribosome, so that's why it's called the ribosomal RNA. tRNA's main function is to carry the amino acid during the process of translation. mRNA is the messenger RNA which is transcripted from the template strand of the DNA in protein synthesis. The sugar is a ribose sugar. The formula will be C5H10O5. If it is deoxyribose sugar, it will be C5H10O4. One oxygen less, that's why it's called deoxyribose. This is called ribose sugar. N2 bases in RNA will be little bit different. Instead of thymine, you have uracil. Like thymine is not present in the RNA. Instead, that pyrimidine base is substituted by uracil. So AUCG is adenine, uracil, cytosine and guanine. This is all about the structure of the chromosome. I have told in little bit detail, that much may not be needed, and go through the chapter. If you have any question, you can put up the question. Thank you very much and be safe, be at home.